My name's Ian Shannon. I'm a graduate student in Dr. Rich's lab. I've been working in this lab since January as an undergraduate from last year. My role in this lab this summer has been partially fish care and mostly molecular work, which includes RNA isolation, polymerase chain reaction to test the efficacy of our morpholino injections. This is done by isolating RNA from our fish samples as they grow up at different time points, then taking that messenger RNA and doing PCR to amplify it. Then we run it on a gel, and that gives us a visual representation of how well the morpholino is knocking down our RNA samples. The first step in our experiment starts here. This is our fish room where we keep all of our experimental fish. This rack here is a system that runs water, keeps them healthy. We come in, feed them 3 times a day. On the top are all of our breeding pairs, and what we do is we select breeding pairs we want every week for injections. We put them in a small breeder box, which is a small box, holds about 1 liter of water with a rough bottom with holes in it. When the fish are in that breeder box, they come together and they mate, they lay eggs. We then harvest those eggs with a screen. We take them back to our lab and we do our injections and our experiments. A large part of the process for what we're doing is measuring how well our morpholino is working. We do this by isolating messenger RNA from samples of tissue. We isolate it using a chemical that breaks down cell walls and then binds to messenger RNA to stop it from degrading. From that, we mix it with some more chemicals that form a cDNA template. After we've isolated messenger RNA, made our template cDNA, we can take that template cDNA and do polymerase chain reaction. That involves taking a small segment of that DNA and making millions and millions of copies called amplicons. And we take those copies into a small fluid like this, and we put it into a gel. And we run electricity through the gel, and it causes those little pieces of DNA to move through it. We can then image the gel, and it will show us if we have the pieces of messenger RNA that we were looking for originally.